Assalamualaikum and very good day to the Azania. I am Nushazan bin Azhar and today I will present a poster of my research project regarding to the recent development in phytoremediation for aquaculture wastewater from seawater. This project is guided by my supervisor IR Normaliza Ahmad and my co-supervisor Professor Siti Rozaimah Sheikh Abdullah from UKM Bangi. This is a picture of my overall poster that I did. Okay, now moving on to the content of the poster. Firstly, starting with the abstract of my research paper. Globally, the aquaculture sector has grown in recent years due to the increasing demand of aquaculture products and the need for more food. This development creates profit and revenue, but it also poses significant environmental issues such as pollution. Dissolved organic carbon, nitrogenous chemical, and phosphorus are typically found in the aquaculture wastewater. The release of a large amount of aquaculture effluent into the seawater results in a rapid increase in a seawater nutrients and causes harm to variety of water species. It also driving to excessive phytoplankton development, which decreases dissolved oxygen levels and leads to the creation of bottom sediments. Phytoremediation is a remediation method used to clean up areas that are contaminated with organic and inorganic pollutants. It is one of biological wastewater treatment approaches that uses the concept of plant-based system. And then toxin in a wastewater can now be eliminated in a safe, low cost, and long-term way rather than by encapsulating or moving the problem to another location. This overview aims to identify and describe the development in aquaculture wastewater phytoremediation from seawater. Additionally, the characteristic of plants used in a seawater phytoremediation process are specified. Okay, next, for the objective of the research, in this study, the objective that were to evaluate are to identify the recent development in phytoremediation in aquaculture wastewater from seawater. And next one is to recommend the use of phytoremediation process in the most of wastewater treatment in the future. Next, for the problem statement, freshwater aquaculture wastewater phytoremediation is the primary focus of majority of recent research nowadays. Therefore, aquaculture wastewater phytoremediation from seawater and suitable plants for the process that can withstand high salinity of seawater will be examined in this study. Next, the advantages and disadvantages of phytoremediation in seawater were analyzed in depth. For aquaculture effluent that comes from seawater, more phytoremediation procedures need to be implemented to the future environment. Okay, move to the scope of study. This study is focused on the advantages and phytoremediation for aquaculture wastewater from seawater. Then, the other scope of research is the determination suitable plant for phytoremediation process in high percentage of salinity system, which is seawater aquaculture wastewater. And lastly, the challenges of the phytoremediation process in aquaculture wastewater from seawater is the last scope of study in this research project. Then, for the method of study, the procedure on reviewing this research project is derived from previous past research paper written by other researchers who were conducting study on phytoremediation process on various aquaculture wastewater. This is a process flow chart of sequence in methodology during the research progress. The The first step is by data collection of study. The data collection refers to the process gathering, quantifying, and evaluating correct insight for the research purpose. And then the techniques of conducting the research and collecting the materials were gathered from the associated electronic database such as Google Scholar, Science Direct, Web of Science, and Scopus. The second step is comparison of research design. This comparison of research design is carried out in a scientific manner with more than one variable being taken into consideration. Next is the analysis of research variables. In order to determine whether or not phytoremediation treatment is effective on aquaculture wastewater from seawater, the research variables will be compared after the design of the study has been determined. Lastly, conclusion. Each component will be explored in depth in order to reach the conclusion. Okay, next, for the discussion, there were four main content that have been discussed, which are nitrogen removal, phosphorus removal, water quality after phytoremediation, and specific plan for the phytoremediation in seawater aquaculture effluent. For the nitrogen removal, in the feeds of all aquatic species, nitrogen and phosphorus were ample. The nitrogen composition of protein ranged from 13 to 19% with the animal proteins containing about 60% of nitrogen. The estimated nitrogen retention efficiency of numerous fish species is 30%. Therefore, some quantity of nitrogen needs to eliminate from the effluent. Based on the research studies by Nizam and others on 2020, there were 5 aquatic plants used on the experiment, which are Centella asiatica, Alpomia aquatica, Salvinia molesta, Ecornia crapsis, and Pistia stratoi to remove ammonical nitrogen in the wastewater. From all of these 5 plants, C. asiatica lowered the ammonical nitrogen by 98%. This is due to its high oxygen transfer abilities, allowing them to create habitats appropriate for the bacterial nitrification while also improving the removal of nitrogen, ammonical nitrogen. Next, in the phosphorus removal, the predominant sources of phosphorus in aquaculture wastewater is phosphorus from aquatic animal diets. Excess dietary phosphorus and indigestible dietary phosphorus are always expelled and contaminating the environment and frequently violating environmental standards. From SD and Hari Kumar on 2019 research studies reported that Acrosticum aurum absorbed phosphate in the effluent. The phosphate concentration in the effluent media during the entire experiment fell to the lower levels during the phytoremediation process. Meanwhile, Ng and Chan on 2021 reported that the use of 3 micropipes system in the removal of phosphate in the aquaculture wastewater from fish farms. The micropipes system are the S. polyriza monoculture system, Lemna species monoculture system, and S. polyriza plus Lemna species polyculture system. This research study shows that the plants use the phytoremediation process can effectively remove the phosphate content in the aquaculture wastewater. Then, the third content is water quality after phytoremediation. The outcomes of phytoremediation on the water quality of wastewater from aquaculture can either improve or deteriorate the water quality. For the COD, which is chemical oxygen demand, based on the Prakash and others on 2018, Eclipsis plant reduced the greatest level of COD in the effluent. Other than that, SD and Harikumar have found that Acrosticum aurum has steadily decreased the COD in each effluent media, but at the moderate phase. For the total suspended solid, according to the Nizam and others on 2020, these stereotypes lower the total suspended solid with the reduced rate up to 98% due to its various root. Over this, root system of aquatic plants or micropipes enhance their capacity to trap and draw particles, which is total suspended solid. Okay, for the last discussion is a specific plant used for the phytoremediation aquaculture wastewater. There are two plants which are Acrosticum aurum and Encornia crapsis. First is Acrostica aurum. Acrostica aurum are the only ferns that belongs to the mangrove group. It has ability in absorbing phosphate to grow. Then it also a salt tolerant plant whose leaves store salt. The plant also decrease the nitrate concentration, increase the dissolved oxygen. And lastly, decrease the BOD and COD levels of aquaculture effluent. The second plant is Encornia crapsis. It potentially in reducing the levels of nitrogen, phosphorus, and heavy metals in the water. Then it reduces the variation in pH, DO, dissolved oxygen, COD, and total nitrogen. Lastly, its root structure provide a large surface area to facilitate physical, chemical, and microbiological processes for nutrient absorption and nutrification. For the conclusion, the technology of phytoremediation of aquaculture wastewater has the ability to eliminate the excessive amounts of nutrients like nitrogen and phosphorus that are present in the effluent. Aside from that, it has a potential to increase the quality of water in the effluent. As the plant for the phytoremediation process can take up the chemical oxygen demand and the total suspended solid, the amount of dissolved oxygen in the effluent will increase as the plant grows. There were certain plants that were utilized for the phytoremediation treatment in the aquaculture wastewater that was derived from seawater. Acrosticum aurum and Encornia crapsis are two examples of plants that are used for the phytoremediation in the seawater. Next, as the recommendation, phytoremediation process need to implemented to the aquaculture wastewater as it is a unique strategy to environmental pollution treatment. Phytoremediation is anticipated to resolve all current crises with the more efficiency, less environmental impact, and economical feasibility. Other than that, more specific plans need to be experimented to know whether it is suitable to use for aquaculture wastewater from seawater. Lastly, in the acknowledgement, I would like to express my sincere gratitude to my supervisor, IR Normaliza Ahmad, and my co-supervisor, Professor Siti Rozaima Sheikh Abdullah, for their assistance in making this work possible. At the bottom of the poster, it shows some reference that I used for this research project. I think I will end my presentation. That's all. Thank you.